When you're learning a new skill for the first time, it makes sense to copy someone who knows what they're doing. A teacher, student in the year above, or a YouTube video. And watch the skill being demonstrated right in front of you. When it's your turn to try it out, it will look and feel different. It's not enough to copy the perfect version of events that someone else has laid out for you. You have to know all the ways something can go wrong so you can reverse engineer and troubleshoot any situation. Hi everyone, I'm Jack Wang, a microbiologist and science educator, and on this channel we talk about using science education to develop transferable skills that can move your career forward in any field. Laboratory training is a rare, expensive, and time-consuming process, especially in research and development or R&D. You're not just repeating someone else's perfected protocol, it is up to you to design all the steps and make sure they are foolproof. Troubleshooting experiments is a complicated topic, and over the next few videos, we will break this down for different lab techniques. Today, the focus will be on antibiotic sensitivity testing, the experiments that flag any new superbugs that will cause havoc in the population because none of the available drugs we have can kill them. So let's imagine that we have isolated a new bacteria and want to test to see which antibiotics will work against it. We've talked about this before on the channel, disc diffusion methods, where we spread the microbe across the entire surface of the plate, add a series of antibiotic discs, incubate the plates, then measure the zone of inhibition around each disc. The bigger the zone, the better the antibiotic works in killing the bacteria. If this zone is smaller than a standardized value determined by diagnostic labs across the world, we consider the bug to be resistant to the antibiotic. Let's pause here for a second and look at three design parameters. 1. How do we know how long to incubate the plates for? 2. Each of the antibiotics added is at a given concentration. How does scientists figure out that this was the best concentration to use? 3. The standardized value that determines if a zone is resistant or sensitive to an antibiotic. How is this determined? The truth is, all of these parameters weren't landed on by luck. They were systematically tested again and again until the most optimal version of the experiment was figured out. To demonstrate how scientists troubleshooted their way into the best version of this protocol, let's use a lab data simulator to recreate these experiments. First, how long should the plates be incubated for? We can pick the type of agar plate, the bacteria, and choose the same antibiotic disc and concentration every single time. Let's set up a series of disc diffusion assays where the only variable that changes is the incubation time. We can pick a series of time points, let's say 6, 12, 24, 48. We incubate the plates using the simulator, where time can be sped up thankfully, and look at the results. We can see that in the earlier time points, no zone of inhibition has appeared. It's not until midway through the time series where we see the zone of inhibition around the antibiotic disc. If you push further into the time series, you can see that bacterial growth and the size of the zone of inhibition doesn't change much. We're past the point of diminishing returns. Ideally, you want the experiment to be done in the shortest amount of time that still gives you valid results. The next point is how reproducible these results are. We set up 3 replicates and we can measure the zones of inhibition present for each repeat and assess how much variability there is plate to plate. You can plot this out using standard deviation or standard error of the mean and decide what is a reasonable amount of acceptable variability, say plus or minus 5 to 10%. How about what concentration of antibiotics to use? It's hard to monitor more than one variable at a time, so let's say the last series of experiments told us the best time point to use. And now everything will be done using one time point. This time the variable will be the concentration of antibiotic. We can try a few different concentrations and see how this affects the size of the zone and also how reproducible it is. If it's too low and there's no zones to measure, or too high and it kills all the bacteria on the plate, either way, it's not that useful for the disc diffusion assay because it relies on a measurable zone being present. Of course, if we're doing turbidity testing of MIC or MBC, minimum inhibitory concentration, or minimum bactericidal concentration, that's less of an issue because you're looking for the presence or absence of cloudiness in a tube, which is measurable. Lastly, once we measure the zone of inhibition and compare it against the set threshold for resistance or sensitivity, this begs the question: who decided these thresholds? It takes a lot more work than 3 repeats to figure this out. At least thousands of repeats in labs all across the world using the same reference strains of microbes were needed to establish these guidelines. This isn't work that a single scientist can do, and it is organizations like UCAS that generate guidelines that are regularly updated to be able to identify the next wave of resistant superbugs. In true higher order learning, it's not enough to copy the perfect version of what others have done. You have to make your own mistakes and pick up the pieces to construct new ways of doing things. Today we talk through optimizing 3 different parameters in one type of experiment, and hopefully you can appreciate that this is just the tip of the iceberg. Once you've done this for one lab technique, we can move on to the next one. and let's talk about how other lab techniques can be optimized or troubleshooted in our next video. This is the Biolab Collective. I'm Jack Wayne. See you next time.